Hello, we're glad you joined us for this live webinar, Metagenomics and Base Modifications from Long, Short, and Linked Reads. I'm Alexis Corrales of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Zymo Research Corporation. Zymo Research Corporation, based in Irvine, California, has been providing the scientific community with comprehensive nucleic acid solutions for DNA and RNA research in epigenetics for more than 2 decades. The company's unique achievements include development of the first available microelution spin column technology for nucleic acid purification. In 2016, Zymo Research released their ZymoBIOMICS line of products to help optimize workflows for microbiomics and metagenomics analysis. Their innovative mock microbial community standards were developed to validate microbiomic workflows to ensure accurate community profiling. To date, Zymo maintains the most comprehensive products and services for microbiome analysis. To learn more about Zymo Research and ZymoBIOMICS, please visit www.zymoresearch.com/ZymoBIOMICS. Let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process obtaining your credits. I will now present today's speaker, Dr. Christopher Mason, Associate Professor, Weill Cornell Medicine, World Quant, Scholar and Director, WQ Initiative for Quantitative Prediction, and Dr. Xiquan Tang, Scientist, Zymo Research Corporation. Dr. Christopher Mason completed his dual BS in Genetics and Biochemistry in 2001 from University of Wisconsin-Madison, his PhD in Genetics in 2006 from Yale University, and then completed postdoctoral training in clinical genetics in 2009 at Yale Medical School, and a joint postdoctoral fellow of genomics, ethics, and law at Yale Law School in 2009. He is currently an associate professor at Weill Cornell Medicine with appointments at the Tri-Institutional Program on Computational Biology and Medicine, the Mayer Cancer Center, and the Field Family Brain and Mind Research Institute. The Mason Laboratory develops and deploys new biochemical and computational methods in functional genomics to elucidate the genetic basis of human disease and human physiology. We create and explore novel techniques in next-generation sequencing and algorithms for tumor evolution, genome evolution, DNA and RNA modifications, and genome-epigenome engineering. We also work closely with NIST, FDA, to build international standards for these methods to ensure clinical quality genome measurements editing. We also work with NASA to build integrated molecular portraits of genomes, epigenomes, transcriptomes, and metagenomes for astronauts, which establish the molecular foundations and genetic defenses for enabling long-term human space travel. Dr. Xiquan Tang is currently a research scientist at Zymo Research. He received his PhD in the Department of Chemical Engineering and Applied Chemistry from the University of Toronto. Dr. Tang joined Zymo in research in 2014 and has since been instrumental in the research and development of Zymo's microbiomics and metagenomics program. His past experiences include work in the fields of environmental microbiology and biological process engineering with an emphasis on bioinformatics, metagenomics, and anaerobic cultivation. Most recently, Dr. Tang has been involved in the development of Zymo Research's microbiomics portfolio. He has multiple publications in different journals, including Frontiers in Microbiology, Applied and Environmental Microbiology, and Philosophical Transactions of the Royal Society of London, Series B, Biological Science. For a full biography on our speakers, please go to the Speaker Biographies tab. Dr. Mason and Dr. Tang will now begin their presentations.
Hello, everyone, and thanks for joining the seminar today, where I'm going to describe some of the work in our laboratory and with collaborating groups on developing standards and tests and new methods for metagenomics detection and for detecting base modifications, which can impact not only work we're doing here, but of course, at the very end, there's some exciting things we'll show about using some of these methods and standards for places as far away as, say, the space station, which is sitting up above Earth. So first, I'll start with a bit of background of why some of this work is important, is that if you don't have good standards for anything really that you're doing, you can't really measure things well, or after where you're measuring things, can you build them well? And so I'll talk about both these things today. Is that an analogy would be, imagine if you didn't have the same distance between threads on screws, you couldn't build buildings or do a lot of construction. And this was true 200 or 300 years ago. It took a while for there to be actual standards, but once they were created, it allowed a lot more comparability and uniformity about construction, and also new things you could just actually build. And so some of the background, this kind of quest to have better standards and metrics has been borne out by editorials from Nature Biotechnology to say that we need, better ways to characterize some of the newer technology, particularly in sequencing. And we've published a lot of work on RNA sequencing to look at what are the standards for and best practices across platforms. In this case, you can see here some platforms for RNA sequencing and now more subsequently nanopore-based sequencing. And there's a lot of excitement because you can not only sequence RNA, but of course you can have a $1,000 genome. There's been a lot of excitement also that this is, as of last year, Illumina claiming you can get a $100 genome sometime soon. So it's definitely an extraordinary time in genetics and genomics. But the challenge is that, we need more and more benchmarks to make sure we're doing it right. So the update to the FDA study is called the SEQC2, which is doing a lot of benchmarking on human samples. You can see here some of the testing designs ongoing for these samples. There's also an interesting way that these cell lines are being blended together. So it's 10 different individuals then pooled. And so we can see a pretty large allelic range that we can test with some of these standards. But that's just a quick update on the DNA standards. But one interesting thing I want to go back to is from the RNA sequencing data sets is we noticed that even when you sequence things really deep, like say to 12 billion reads, we can actually see that we never really reach a real plateau. We keep finding new transcripts, new isoforms at really low levels. You can see on the figure on the right, and also it did depend on which gene annotation used. And that was true for RNA sequencing, and we know it'll be true also for some work in DNA sequencing and panels and rare mutations. But what hadn't really been addressed then, but is beginning to be addressed now, is the fact that there is more than one genome, of course, in our bodies. Of course, there's been a debate about exactly what percentage of cells are you in terms of bacterial versus human, but we know there's a lot of them, probably about at least half of your body's bacterial cells, and you can see a lot of debate in some of the literature. But it means really that there's a lot. So if you were to do this effectively and look at this, from the view of the microbiome, you could see, you can see here that the human cells are very large, but you're covered and generally, awash in these bacterial cells. And if you step onto a scale, it's about a few pounds of your body is actually bacterial cells, so you can legitimately blame them if your diet is not working as much as you'd like. The other thing is the great work done by a lot of people in the field, like Curtis Huttenauer and Rob Knight, is to define sort of the commensals in the normal microbiome from the Human Microbiome Project. But a lot of this was, defining these just normal ecosystems, which are important outside the body, but also inside the body. We know, for example, that about a third of the small molecules in your blood are either made by or trafficked through the microbiome. And this has important implications also for how you look at human health, so including obesity or how you process food or inflammatory bowel disease or even other diseases that are related even to mental disorders. You can see a list on the right of things that have been linked to the microbiome to date. So it's a really extraordinary time, and the microbiome is obviously very important because you want to get, if you have it right, a sort of microbiome force field rather than something that might be hurting you. It can actually really give you a lot of beneficial sort of protective abilities for outcompeting other bacteria. And one great example of this is fecal microbiome transplants, where you can actually transplant an entire ecosystem for someone who has a really reticent infection that cannot be treated well with antibiotics. In this case, you can see there's been a lot of people even starting to take matters into their own hands with community action groups like thepowerofpoop.com, which is I couldn't make it up if I tried. It's an actual real website where people doing things, doing transplants on their own. But it's not because they're crazy, it's because they're looking for something that actually works well. And so you can see here, success rate for C. diff treatment is actually pretty high. And so far, one good thing is that it's been regulated more as a tissue than a drug, which is helping some of the transplants. But here too, even with all this promise and power, the question is, well, if I do a transplant, how do I know that it'll work or what I am transplanting? Has historically depended a lot on 16S ribosomal RNA sequencing. And you can see here from work from Gail Rosen and Hirschberg is that if you say, well, if we were to pick a gene today to define what's in a sample, where would it fall if you pick different marker genes that are present in almost all bacteria that you can amplify, and you compare to, say, amino acid identity or compare to, say, general similarity of distantly related prokaryotes or closely related ones, you can see 16S RNA is really, you wouldn't pick it today if you were to pick again from scratch a marker gene that you could use in bacteria and archaea. It's an okay marker for distantly related prokaryotes, but not really at all for closely related ones. And you can see here some examples where you have problems with the phylogeny defined by 16S. And so it seems, I think it was really just a useful accident of history that we could define ecosystems through the lens of one gene. But we know now that, there's many other things you can see from shotgun sequencing, which we prefer for all of these reasons listed here, as things you can start to pull out from the samples. It's a bit more expensive, but that cost is coming down. And we've already seen some utility in the fact that if you do shotgun sequencing of all the isolates, say, from a clinical and ICU, you can see here that what you normally think of through culturing, you might have one kind of strain. You actually get many more stratification of what you've detected from a clinical sample. And so, which is exciting, but still it comes back to the original question is, well, are we any good at measuring, and can we really reach what I'd like to say, I guess, is precision metagenomics, that you don't necessarily know where any sample came from, but you want to make sure that you've measured it correctly and that you know what is there. And so one interesting chart we've made, and it just published this in a paper I'll talk about in a few slides, is if you can change how deeply you sequence a sample and then you subsample, you'll keep finding new species as you sequence deeper, very similar to the chart that we made for the FDA first study in 2014. And what's interesting, though, is if you do it based on pathway analysis of what's present, say, from human pathways, that does, it basically give you some more consistency as a function of depth, but it's still not perfect. And so this has led to some work through NIST, which is called the Microbial Standards Group in the International Metagenomics and Microbiome Standards Alliance, or IMSA. And to date, we know there's at least 71 computational tools for profiling microbial communities. So we decided to do a little bit of a comparison with this paper we just published last fall, which is looking at, depending on what your goal is, are you looking for sensitivity or specificity? What are your key metrics you're looking to measure these classifiers based on? This gives you kind of a chart here. But one of the take-home points is that across many of the tools we looked at, if you look at the genus level, it's not too bad. But if you think, say, it's species, or you go down to even subspecies level, the strain level identification is still one of the really hardest challenges from shotgun sequence data, but is also where the most important factors are to be measured. So this is, while the most difficult, perhaps still one of the most important. And so one of the reasons that we published this paper was somewhat just for our own erudition to know what to do, but also we wanted this as a place to go for people who are looking for good standards and benchmarks and well curated and annotated and titrated, if possible, data sets and samples. And moreover, that we don't just want to sequence them, we want other people to order them, not just today, but also in the future. And so one of the data sets in here, if you go, if you look at one of the challenges, one of the more complex data sets, we saw 4 log 10 differences in the number of predicted species from one of the complex samples. But there is no standard for a place like this, and there is no really standard for really a complex situation like this, for example. But we did, wherever you're looking in really strange places like this is the Gowanus Canal Superfund site, you need to know that you at least have titrated positive controls, and that's one of the reasons in that paper, the biopool is the name of the file, but it's actually the ZymoBIOMICS control. That was their early name of the product, and it is actually now a standard product. And so when looking in really strange places, we want standards. That includes, for example, like subways, which I'll talk about briefly. But we really have a question of who's there and what are they making. And to do this, like any good experiment, you do need some control. So to that end, I want to describe some of the work that we've done, and a lot of this is in very close concert with Zymo. When I met some of the people from Zymo saying they wanted to develop a titrated standard of bacteria and yeast, I thought, well, that's fantastic, because it really either didn't exist, or what was out in the field, you could only order a very limited amount of it. So, to give a quick snapshot of what's out there, this is, again, a very focused effort from a group at NIST and several academic and industry groups working together to develop these standards. There's more than one, just to be fair. There is one from ATCC that was released, which has been characterized, and we know that essentially if you change what primer that you use, you can see different proportions of genera, depending on which region of the 16S you look at, so we know it's very important. And ZymoBIOMICS, actually, when they released their standard, this was, I think to my knowledge, the first really multi-kingdom standard, so both bacteria as well as yeast that you can see here, put together different GC, different abundances protein that are titrated together so you can utilize. And we've been using this for now almost 3 years because we can use it. We've been using it in almost all our experiments because it's really, like any good experiment, you will need a positive control. And so we've seen, we've published one paper on this. We're looking at different numbers of cycles of PCR to test on different, for example, in this case, QIAGEN or Illumina library prep. So this gives us, this is analyzed with one codex here, you can see, and it really gave us an immediate readout of, is it working? Are we doing well? Does the data look about right? And so in this case, we could see very much what we were expecting to see, the Cryptococcus and the Saccharomyces, at low abundance, and the other ones at generally equal abundance. Again, not perfect, but it gives us a sense of what's the technical noise at the bench, what's the noise at the computational side, and anything in between. And so we can see here that PCR was not our big variable. We do know, again, we can see these species pop up, but we know that also different protocols, and this came up a little bit from work that Dr. Tang has done, is that different protocols we know will give different yields of what should be theoretical output from some of these organisms. So this is another reason to have the control, not only in a DNA form, but also in a cell form, so you can do this kind of testing. And so this is another reason to have these controls. And so we, as I mentioned before, all these data that we put out there from the paper from Alexa McIntyre, who's a grad student in the lab, they're all publicly available. They're there for people to use for their own testing of algorithms or methods, and really there because mostly built because I wish they had existed when I started working a lot in metagenomics, and so it's the first beginning step to have some of these standards out there. One other note is that we did also just release a preprint, so this is 10x data, so chromium data using the Zymo controls, so they're pooled together, and the data here are publicly available. We've just put in a preprint showing you can actually use 10x reads to generate these read clouds to try and stitch together in each individual molecule which species it came from, and we've also released code called Minerva, which is open source and publicly available from David Denko, who's a grad student in the lab, to do deconvolution of these long reads that are synthetic long reads from the molecular barcodes. And so that's, again, available for people to use. So what we're doing with all this, so that's kind of a bit of the backdrop. I'll tell you a bit about what we've been doing. So the big question is, what have we been missing? One of our big questions in the lab is what have we been missing, for example, what's in the urban microbiome, or what the New Yorker called the metropolome. But in some work we've done, this is looking at microbiome of cities. This was also announced as part of the National Microbiome Initiative and is sponsored by the Bill and Melinda Gates Foundation. But it's a project we have called Metasub, which we build these genetic maps of the Earth cities as a way to track antimicrobial resistance and also to mine for new biosynthetic gene clusters. And in every city, we use the controls of Zymo for the positive controls during sampling and processing for all these samples. So what this looks like is, I don't know if the video here renders, but it's a planet that you can see. What this means is we've been sampling since 2015, basically sampling around the world to generate sort of a global map of the Earth cities and what's present metagenomics-wise. So this ends up looking, it looks like a lot of shipping. We do a lot of this every summer on the summer solstice. We do a global city sampling day. You can see here is the samples leaving lab to head many places, like to Marseille, France, to Tokyo, Japan. You can see here some of the collection tweets, which are very fun. A lot of people, after sampling, end up drinking beer at the end of the day. We find this to be very common metasub occurrence. We have teams in Singapore, Oslo doing also air sampling, San Francisco working with Jessica Green, in Bogota, you can see here, of course, in New York City, and then in Kiev, and then Lima, and you can see here Minneapolis with some Vikings, and also in Denver. And then finally, other places like Fairbanks and Doha, you can see. And then this year we got the first mayor home. We had a city council members helping us in New York City, but we got Catherine Pugh, the mayor of Baltimore, actually out swabbing this past summer, which is really fun. You can see her appropriate perplexed-looking face there during the sampling. But again, in all cases, Zymo controls were behind the efforts to do the probing. And so One thing we've seen so far, for example, is that there's a really wide diversity of antimicrobial resistance markers, particularly in New York City, and we're expanding this analysis to the other cities. And we can see so far that there is some stratification of different cities around the world, is what we can see for both their methyltransferases that are present, but also just their AMR markers that we can see. You can see different cities around the world here colored do show not only some degree of clustering based on geography, but also we can see different highly abundant antimicrobial resistance markers in different areas of the world, which is something we're using to track when and where the most abundant ones are appearing and then how they're moving. And also, we have seen that this data has been useful not only for our sake, but also other groups have been downloading our publicly available data to expand out the tree of life to discover new branches on the tree of life, which we find really exciting. So one other idea we had is, what if we kept doing this not just around cities, but if we started doing it, for example, at conferences? So recently at the ABRF meeting this past year, we actually swabbed everyone's phones and basically wanted to see what would happen on people's phones if you were at a conference. So this involves setting up a sampling basically paradigm where we didn't get much sleep. We'd collect samples at the meeting, get them over to Luminous headquarters, do a library prep very quickly, do some quick analysis, and then want to make a report at the meeting 36 hours later so that everyone could look at their sample. Not surprisingly, we used the positive controls from Zymo during this experiment to make sure we were seeing pretty good controls, which indeed we did here. And we really use this as a way to generate hypotheses about what people were doing at the meeting, kind of where they had been. So, like, for example, could it reveal what you've been eating or doing? And here we think that we saw a lot of human DNA and some unknown, but a lot of bacterial. You can see here's our two positive controls. But some people had really high amounts of unknown DNA. One person maybe appeared to be alien-ish because we couldn't detect a lot of their DNA, didn't match anything known. But we did see other interesting trends. For example, across food and pets, we saw some things pop up from people and then wanted to say, well, is that really what it's matching? And so we asked this person if he'd eaten an orange, and he said it's okay to reveal his name. He said, yes, indeed. I just peeled and ate an orange before he gave us his phone. One person looked as if he'd had maybe pork or bacon, and indeed this is a guy who had just used the phone to take a picture of a pulled pork sandwich he ate the night before that was still apparently on his phone. We also then looked at other comparisons of what is the cosine similarity to known body sites, and shoes look the least like skin, but a lot of other samples do look like skin. And we could also see a little bit of a signal for people who say they have cats, and for people who had dogs, a much stronger signal that was present. So it kind of serves as a forensic tool as well as a discovery tool from the shotgun sequencing, which was kind of fun. And of course, we saw antibiotic resistance present in a lot of people's phones, which is we find pretty commonly in a lot of places. And also one last bit of hypothesis generation is that we could see, basically people who had just taken antibiotics had a pretty different profile from those who didn't, and maybe enriched for a Haemophilus organism, which we thought was kind of an interesting hypothesis from the data. So that's where we've been doing it in sort of sequencing everywhere on Earth. But where else could we sequence? One of the last projects I want to tell everyone about is two specific projects that we're doing in space. So one is doing participatory medicine with twin astronauts that we've been studying for the past few years, the Kelly twins. In this case, we're trying to understand the impact of space travel on the human body as well as the microbiome in what's called the NASA Twin Study. And so a lot of these results are embargoed until a few weeks from now, but there'll be more data coming out soon once the papers come out. But we do see some changes in space, as you can imagine, in the microbiome. But one thing that we've just published that I'm happy to tell you about today is a lot of work we did with Aaron Burton and Sarah Castro-Wallace at JSC, Johnson Space Center, and others at NASA to think, well, could we sequence in space, and could we get these standards to work in space? We actually, of course, NASA, when you start a project, they say you need a patch. As we were planning the mission, we had an issue where one of our resupply rockets exploded on the way to the space station. So we got this, though, really nice letter saying, sorry that we blew up your supplies, and we're going to urge you to continue your inquiry and make sure that we get more supplies up there, which I thought was very touching, actually. And as we had a chance to do work on the parabolic flight simulator, we could actually see, would the nanopore sequencing work in zero gravity? And this is a video of actually Andy Feinberg doing testing in zero gravity, which was very difficult, but I can tell you did eventually work. We actually were able to load a live around of the flow cell, and it showed that it was possible to sequence in zero gravity. And this basically also led to a new algorithm to analyze the data, and eventually was published last year. And then right around the same time, Dr. Kate Rubins was getting ready to go up. She's a trained virologist to go do the first sequencing in space. She made it up there, no problem. And also we know the resupply mission worked well. Eventually, we got actually the first sequencing in space to work for the very first time. And we got not just one run, but we got dozens of runs to complete what some called the dawn of genomics in space, and then also got out to 1 billion bases sequenced. So one really exciting thing is not only did it work, it actually worked really, really well. We could see, at the time at least, this was very good data. We could see in space, in zero gravity, from E. coli and mouse and lambda DNA, and looked very, very good. And we got even as good or better data in space than we did on Earth for the same control library. So it looked really, really good. And we just published this at the end of December, sort of the first genome sequenced and then assembled offer that you can see here from work that we did with collaborators at NASA. And one really exciting thing that happened as a consequence of this is when they started thinking about future missions after we were finishing the data there, NASA was saying, well, we need more controls. And so actually just a few weeks ago, they were able to use nanopore sequencing and actually doing library prep in space. And the controls that we used during the development of this were indeed the Zymo controls. So they have made their way up to the space station and are now truly kind of both earthbound and spacebound or space flying controls, which we are using not only for some of these studies, but also an ongoing work for the SEQC2 now includes some of the Zymo controls that we're using to characterize what's present in them. So to this end, we've collaborated not just with the FDA, with the SEQC group, but also with the Argos, which is the reference grade bacterial strain group led by Heidi Schittig. And you can see here we've been sequencing on PacBio and Oxford Nanopore each of the individual strains from Zymo, and we're almost done with the final base modification detection, and we'll be releasing all these data very shortly, again, in close collaboration with Zymo to just establish these controls. And so I can give you a little bit of a preview of what we've seen so far. So in particular, as many of you probably know, methyl-6A is a very common modification of adenosine found especially in bacteria. This is one of the first data sets that looked at this. And we've developed a base caller for nanopore sequencing to call epigenetic modifications in nanopore data. And in particular, what we look at is the current information, the deviation from expected current. This is the training run versus the test run for an algorithm called mcaller. And you can see certain positions as they move through the pore are more informative than others. So, for example, the fifth position you can see here is much more informative to differentiate M6A versus A from our data. And we've tried a variety of different machine learning methods to see which ones would be the most effective and gotten out to about 94% accuracy with some of the methods that we've tested. And what's really exciting is when we started comparing these to PacBio data, we could see there are known motifs for where you should see base modifications in these reference strains. And we can see for PacBio versus Oxford Nanopore using the Zymo strains that we can pick up some interesting, new motifs that are present in Oxford Nanopore data that we didn't quite pick up from the PacBio data, where it looks like, it does have a signal where you can see the canonical motif versus not, where you get the quality value for the base modifications, but the signal seems a bit stronger for the Oxford Nanopore data, at least for these sites so far. So it's interesting having these standards because now we can have two different metrics to find, in principle, all the base modifications that are present in these Zymo controls. The next thing we're doing, again, these data will be released relatively soon. We're just finishing some of the latest version of the base modification caller from PacBio, and then we'll release all these data likely by AGBT or maybe ABRF meeting, so within a couple months. And again, for people to use as a part of their own development for tools and methods. And the long-term view, of course, is not just to measure things, but to then start using them as a way to build things. And so engineering genomes used to be thought as something you couldn't really do that easily or that well, but it's already been done, for human genomes, and human embryos have been edited. Entire, pig systems have been edited so you could grow human transplants in them. This led to a lot of discussion about a synthetic human genome in a project called Genome Project Right, which I'm working with many people listed here, and we're developing ideas around this. But the principle is that understanding biology, I think, is best measured by can you engineer or tweak something and then actually predict the outcome. So we've been talking a lot about even engineering defenses, genetic defenses, to be internal to astronauts, or at least on their microbiome on the outside is another idea that's come up, because they interface, for example, in the space station with a microbiome all the time. It looks like this, it gets moldy. It's just like Earth in that regard, that you have many complex interactions and exchanges of microbes all the time. And so modeling them, measuring them, and eventually even engineering them to be more protective and more commensal-like would potentially be a very good thing for them, especially in a limited environment with limited abilities to counteract something that goes wrong. So that's a bit background on sort of what we're thinking about for NASA, but also, again, these controls we've built, we've been mapping as part of them what are the methyltransferases present in each of the Zymo strains from these controls, and mapping these out. Again, this preprint will go online likely very soon, but we can see it's not just what they have for just essentially genes that are of known function, but all the biochemical capacity of what methyltransferases they have, and how that may or may not impact virulence or even just by adaptation to space-like environments is something we're keeping an eye on for. So in the long run, I think about engineering not just a space station, but even entire planets. Some people at NASA talk a lot about terraforming or going there for long-term missions, and this does seem strange to many people to think that far out, but I think it's actually not that different from what we already do on the first planet of Earth, where we think of planetary scale monitoring and really mitigation of any things like climate change or any issues on the planet and speciation and really conservation. So this is just really the second time I think we're doing it. So in the end, I want to thank you everyone for attending. Also state that life evolves and clonality shifts at genetic and epigenetic layers. We see a lot of skin and unknown DNA in the subway systems in the Metasub project, but we see a lot of potential biosynthetic gene clusters. We can see what you've been doing on your phone. The Zymo controls have been really now globally or even suborbitally, low Earth orbit use controls for all these genetic applications, and we've really enjoyed using them. And also we can actually sequence on the space station. The nanopore sequencer is actually now a validated piece of flight hardware, so if you submit projects to NASA, you can just request to use it, and it is yours to use. I want to thank everyone in the lab who makes all this work possible and really makes it possible for me to tell you about things that we're doing, and great. Thank, of course, the Kelly twins for their blood, sweat, and tears to make this possible. This is their official NASA poster. And, of course, the many collaborators who help with the Metasub project, Consortium for Space Genetics from Harvard, collaborators from JPL and UCSF, and also funding from NIH and Gates Foundation and NASA and WorldQuant and Bailey and several others. And this is thanks to everyone in the lab and collaborators. And thank you very much for your time.
Thank you, Dr. Mason, for that informative presentation. I will now turn it over to Dr. Tang for his presentation.
Hello, everyone. My name is Chen Tang. So I'm a research scientist at Zymo Research. Today, the topic of my presentation is improving the accuracy and reproducibility of microbiome measurements. So this is a brief outline of my presentation. At the beginning, I will talk about the issue of bias in the field of microbiomics. And then I would share some of our experience and also examples about addressing bias in different steps of microbiomic workflow. In the end, I would like to introduce a new product from us. It's actually a new microbiome standard that can help you assess accuracy and also detection limit simultaneously. So as you know, in the past decade, the field of microbiomics has been developed rapidly, and it has greatly changed our understanding about microbiology and also how microbes interact with human beings. And at the beginning of this talk, so I would like to talk about the problem with the bias issue in the field. Actually, right at the beginning of this field has also been criticized for poor data quality and poor data reproducibility. Even the biggest players in the field have reported contradictory results. Here I will give you an example to describe the problem. The first is a story published in Science News of 2014, and the authors split one stool sample into two and submit each of them to each of the two most well-known microbiome profiling organizations, American Gut and UBiome. But surprisingly, the microbiome profile coming back from these two organizations are dramatically different. The profile from American Gut says there's about 60% of bacteroides, while the profile from the UBiome says the bacteroides is only about 30%. And this is the problem we need to overcome. The second example is even more striking. So the Human Microbiome Project and also the MetaHit project are the two biggest projects that have ever conducted to profiling human gut. However, their interpretations of the human gut microbiome are actually quite different. So this figure shows the average profiles of the human gut microbiome determined by these two projects. And as you can see, the two profiles look dramatically different. The question is, are the differences we see here, are these differences real, or are these differences biological differences or technical differences? This is a paper published in 2014. In the paper, the authors compared the DNA extraction kits from MetaHit and HMP, and actually found that the DNA extraction process used in these two projects can actually dramatically change the microbiome profile in the end. So if you know about how microbiome profiling was done, you would not be surprised that there's a huge problem of bias in this field. This is because the microbiomic workflows are complicated. And at each step, bias or artifacts can easily introduce and cause inaccurate microbiome measurements. So this slide shows different factors that can easily introduce into a workflow. And the microbiome measurement is actually required to be both quantitative and sensitive. The ideal microbiome workflow needs to be unbiased and free of contamination. However, this new requirement was not taken into consideration when tools were designed for microbiome research. Because of that, new innovations and new tools need to be utilized to improve the accuracy of microbiome measurements. So next, I will show some examples and experience of how we use some tools to prevent and address bias during sample collection and preservation. As the first step of the microbiomic workflow, sample collection is of critical importance because any potential bias introduced in this step will carry over through the whole pipeline and affect the final results. And as you know, a microbial sample can change easily after sample collection due to undesired microbial growth or a nucleic acid degradation. And in many cases, access to refrigeration or freezing is not available. So there's an urgent need for cold-free and unbiased method to preserve microbiome samples. So this is so we did an experiment. This figure shows how the microbial composition of a feces sample can change when saved at room temperature without protection. And as you can see, the change already happened in just one day. So our solution to prevent bias during the sample collection is a new product called DNA RNA Shield, which when added to a sample can stabilize both DNA and RNA profile at room temperature for more than 1 month. So this figure shows the microbial composition of a feces sample saved in DNA RNA Shield and samples over the course of 1 month. And as you can see, the microbial composition stayed almost constant. If affordable, many people would still choose to use freezing to preserve microbiome samples. However, there's a problem. But however, even with freezing, there's a potential problem that can cause bias. For example, in the literature, there is a recognition about potential bias that can be introduced by freeze-thaw cycling. So we did an experiment to confirm this potential problem. So we took a feces sample and let it go through frequent cycles, 5 cycles, 10 cycles. And as you can see, by as you can see, go through 5 cycles of freeze-thaw cycling and can cause a dramatic change in the microbiome profile. And that is that can go through a dramatic change in microbiome profile. And actually, DNA anesthesia is also a solution to prevent this problem. This profile shows how DNA anesthesia, how samples save in DNA anesthesia and then it go through the cycling process. And you can see in this case, the bacteroides is protected in this case. So next, I will talk about how to potentially address bias during DNA extraction process. The step of DNA extraction has long been known to cause bias during microbiome profiling. So in this case, we're comparing 4 different DNA extraction protocols using the ZymoBIOMICS Microbial Community Standard as a defined input. And these 4 DNA extraction protocols include our ZymoBIOMICS DNA mini prep and 3 other most commonly used protocols that was currently used in the field, including the Human Microbiome Project Fecal DNA Extraction Protocol, the Mobile Pulse Soil Kit, and the Stool DNA Extraction Kit from QIAGEN. And as you can see, compared to the theoretical value, the profile generated using our kits have almost perfect agreement with the theoretical value, while the other 3 kits have dramatic bias. And if you pay more attention, you can see that these 3 other kits overestimate the abundance of easy-to-lyse organisms, such as gram-negative organisms, including Salmonella enterica, E. coli, and Pseudomonas aeruginosa. And they also underestimate the abundance of some tough-to-lyse organisms contained in the standard, mostly gram-negative ones. For example, Listeria monocytogen and Staphylococcus aureus and Enterococcus faecalis. So if you do the same comparison but using a real feces sample, this is how the profile would look like. So if you assume the profile generated using the ZymoBIOMICS DNA mini kit is more close to the real composition of the feces sample, then the other 3 protocols overestimate the abundance of Proteobacterium and Bacteroides, and underestimate the abundance of Firmicutes and Actinobacterium. And coincidentally, Proteobacterium and Bacteroides are mostly gram-negative, easier to lyse, and Firmicutes and Actinobacterium are mostly gram-positive, tough to lyse. So this graph basically shows the same trend as we shown in a comparison using the Zymo controls. So next, I will give a particular example about lysing bacterial endospores. In this case, we look into how mechanical lysis can lyse bacterial endospores. So in this experiment, we use the endospores from the Bacillus subtilis and let it go through mechanical lysis using the ZR bashing beads lysis tube, which has a mixture of 0.1 and 0.5 millimeter beads. And this beading tubes is used as part of the ZymoBIOMICS DNA mini prep. And in the case, so basically the suspension of endospores of Bacillus subtilis goes through mechanical lysis. One with mechanical lysis and the other one without mechanical lysis. The lysis was performed 5 minutes on the FastPrep MP FastPrep 24. And through this experiment, we found that more than 99% of the spores were lysed. However, not all mechanical lysis are built identical. So this figure shows the comparison of our DNA extraction kit with the power soil kit, and you can see the yield, the lysis efficiency, and also subsequently the DNA yield is much lower with the QIAGEN power soil kit. So after this, I will talk about addressing bias during library preparation process. Here I will use the 16S library preparation as an example. The source of artifacts in 16S sequencing include a PCR chimera, reagent process contamination, sample cross-contamination, barcode contamination, MySeq run-to-run carryover, PCR errors, sequencing errors, etc. The defined composition of microbiome standard is really helpful based on our experience to help you identify these artifacts and optimize the library preparation process. So in this case, we're using the ZymoBIOMICS Microbial Community Standard as the defined input. So this figure shows the microbial composition of the ZymoBIOMICS Microbial Community Standard when sequenced and analyzed using a regular workflow. And as many as 43 bacterial genera were identified, while the Microbial Community Standard should only have 8 bacterial genera. So after a series of optimization, this is the profile currently we have when using an optimized workflow, and only 8 bacterial genera were identified, which agrees almost perfectly with the theoretical composition of the standard. Another problem with 16S sequencing is the limited coverage of the common 16S primer researchers commonly use in the field. So we also try to improve the coverage of these primers. So we go ahead and we design the primers based on the most current reference database. So in this case, we optimize the primers, 16S primers for V3 and V4 regions. So as you might know, the common V6, V3, V4 primers only covers the domain of bacteria and has no coverage for either archaea or eukaryotes. And we go and we design the primers and manage to expand the coverage to cover the majority of archaea. And this is the red columns indicates the coverage of our current primers. And at the same time, we also improve the coverage for major phyla, that is for major bacterial phyla as well, which was shown in this figure. So in the end, I would like to introduce a new microbiome standard that we are going to release soon that will help you to assess both the accuracy and detection limits of your NGS workflows. This standard is called ZymoBIOMICS Microbial Community Standard 2 with log distribution. So basically this standard contains 10 different organisms and log distributed abundance. The horizontal axis of this graph shows the theoretical abundance of different organisms contained in the standard. And the vertical line shows the NGS measured abundance. And as you can see, the agreement between the theoretical abundance or the defined abundance with the NGS measured abundance is almost perfect. And this indicates two things. One is the accuracy of the theoretical abundance, and the other one is the accuracy of our NGS measurements. So using this microbiome standard, one can assess the performance of 16S workflow. So in this case, we shows the results when using the standard to assess the workflow of two different 16S workflows. These workflows differ in the bioinformatic analysis. One uses CHIME and the other uses data2. And the conclusion is the result coming from data2 seems to be more accurate as it has no false positives, while the result coming from CHIME has about 9 false positives. However, the result coming from CHIME seems to have higher sensitivity as it was able to detect the presence of Lactobacillus fermentum, while data 2 cannot in this particular case. This standard can also be used to assess the performance of different shotgun metagenomic workflows. So in this case, we compare the performance of 3 different bioinformatics pipelines used for shotgun metagenomics. So as shown in this figure, we found that these three different pipelines have similar detection limit. They all detect down to the organism of Lactobacillus fermentum. But the metaphyllum 2 and the commercial program X have identified some false positives, while the pipeline of MOTU did not find any false positives. Yeah. So in the end, I would like to highlight some of our efforts, which trying to improve the accuracy of the microbiome measurements. And all of these efforts, in the end, generated a complete portfolio for the microbiome measurements. So in the end, I would like to thank all my colleagues from Zymo Research, especially to our director, Dr. Larry Jha, for the R&D department, and our director of the nucleic acid purification department, Ryan Kemp. And thank you, and I can take questions from here.
Thank you, Dr. Mason and Dr. Tang, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question, and click on the Send button. Dr. Mason and Dr. Tang will answer as many questions as time permits. The first question is going to be for Dr. Tang. In the comparison of human gut microbiome profiles between HMP and MetaHit, which profile do you think is more accurate?
Okay, so for this question, I think it's actually difficult to compare 2 different DNA extraction kits using a real feces sample that have unknown composition. With such a comparison, you can only see the profile coming out from the 2 extraction kits are different. Based on what I shown earlier in the presentation, we concluded that the HMP, the Human Microbiome Project DNA extraction protocol, is actually dramatically biased towards easy-to-lyse microbes, such as Bacteroidetes in the gut microbiome. I cannot comment on the performance of the DNA extraction kit from the MetaHit because we did not include that in our study. But the MetaHit protocol seems to be, based on my own opinion, seems to be more accurate, although the workflow seems less streamlined. So I hope that answers your question.
Great. Our second question is going to be for Dr. Mason. How much do long reads versus short reads help for the controls?
Yeah, we've seen that the longer the reads are definitely the better, so you can get much more unambiguous taxonomic assignment when you have the longer reads, whether it's single molecule reads or linked long reads. We've seen a lot better potential in those long reads for mapping. So if we can get them, we prefer them.
Our next question is going to be for Dr. Tang. Why are bacteria more impacted by freeze-thaw cycling than others? How is DNA, RNA field able to protect microbiome profiles from bias caused by freeze-thaw cycling?
So freeze-thaw cycling has long been known to cause damage in cells. In some applications, freeze-thaw cycling is actually a way people use to break down biomass. And we know Bacteroidetes are most mostly gram-negative with fragile cell wall. And I hypothesized that procycline can actually lyse bacteroides cells and release their DNA, which is then degraded by extracellular DNases that are present in feces samples. So DNA RNA shield can actually at least work by protecting the released DNA from degradation of DNases.
And our next question is going to be for Dr. Mason. Do you think more species are needed in future controls?
So I think we've seen a lot of controls for bacteria and also for fungal species, but I think absolutely there's a lot of mystery still on how you get accurate parasite identification or other species. Obviously, plants can be very complicated and even be multiploidy, so you can have octoploid species, which can complicate some of the measurements. So definitely when you get into more broader looks at what's in environmental samples or complex clinical samples, I think more species will be better.
Okay, and Dr. Tang, how do you control and reduce false positive in taxonomy identification?
It's actually very important to understand where the false positive came from. They can come from reagent contaminations, PCR chimera, sequencing errors like index swapping, artifacts or defects in the sequence analysis, and also potential errors in the reference database, etc. So an effective way to help identify and control false positive is actually to use a reliable microbiome standard as a defined input. So you know what goes in, and you expect what goes out.
And Dr. Mason, how about RNA methylation?
Yeah, one thing we only discussed briefly today is that the RNAs and RNA viruses in particular, and actually almost all RNAs we've ever looked at, have differing levels of RNA methylation. So this is another area of future work that I think will be interesting to develop in the microbiome and metagenomic space, or really the metatranscriptomic space. So it's a new area of research that we and a lot of others have started to look at, and it's pretty exciting because a lot is still unknown.
And we do have time for one more question. Dr. Tang, what steps in a microbiomics or metagenomics workflow can affect the detection limit?
I actually think every step in the workflow has the potential to affect the detection limit. And for example, the choice of bioinformatic pipelines affect the detection limit as demonstrated in the presentation earlier. Also, insufficient lysis during DNA extraction can decrease the detection limit. Insufficient sequencing depth or poor sequencing quality can also affect detection limit. So there's a lot of examples like this.
Since there are no more questions, I would like to once again thank Dr. Mason and Dr. Tang for their presentations. Do you have any final comments?
No.
No.
I'd like to thank the audience for joining us today and for their interesting questions. We would like to thank our sponsor, Zymo Research Corporation, for underwriting today's educational webcast. This webcast can be viewed on demand through July 2018. Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.